All right. Hi, everyone. Welcome. We are going to go ahead and get started because it is 5:30. Thank you so much for joining us tonight for our COVID-19 speaker series. I believe this is our 7th one that we've been doing, and we're going to continue to do them throughout the course of 2021. So we are super excited to have Gregg Dean here today. And just while I'm talking, please feel free to take some time, download our free app that we have. It updates you on events that are going on in the area. You can connect your membership to it as well. You can hear about other things that are going on with CSU. And it's just an all-around great app to have, and it's free. We just like to give up free stuff, apparently. So it's all you. Go ahead and use your QR code, or you can look up the CSU alumni app in your store and just download it there. We understand that these are challenging times for people, and we are so excited to be able to offer an educational webinar for all of you. We are here to build RAM community and be a resource, and we hope that this webinar is just one of the many ways that we can do that. I would also like to thank all of our members for participating tonight. Without members, we would not be able to host events like this. We have a lot of members here tonight, so we thank you so much for your membership. If you ever want to know more about membership, I will post something inside the chat in order for you to follow up and hear more about what it is to be a member and how much the cost of a membership is a year. While I'm talking too, please feel free to open up your chat feature. And on the bottom of the screen, there's a little to button that says all panelists. If you want to change that to all panelists and attendees, please feel free to write your name, what year you graduated, and where you're coming from tonight. We get alumni from all over the nation and all over the world every once in a while. So it's really cool to see where our alums are coming and being able to connect people from other areas as well. So please feel free to do that when you get a quick moment. I also want to thank our partnerships for today. We have 3 partners that we work with in order to do the COVID speaker series. Women in Philanthropy has been greatly appreciated with how much they've been pushing information out. The Vice President for Research has been helping us find our speakers. And then the College of Veterinary Medicine and Biomechanical Sciences has been helping us find speakers as well for this event. So we thank our partners for everything that they're doing in order to make sure that this event is gonna happen. I also want to remind everybody that tonight is a webinar, so we cannot see you and we cannot hear you. If you have a question, please feel free to use the Q&A box or use the chat feature. I will be monitoring all of the chat features and the Q&A stuff, and we will answer questions throughout the presentation. So if you have a question, feel free to write it down, and I will stop Gregg from talking, and then we'll answer that question before we move forward. There will be time at the end of the presentation as well to answer questions too, so feel free to ask questions there as well. And now, without further ado, I would like to introduce tonight's guest speaker, Dr. Gregg Dean. Dr. Dean is a 3-time CSU alum, if I'm correct, and also the professor and head of the Department of Microbiology, Immunology, and Pathology at CSU. And I'm sure everyone is probably tired of hearing me speak, so without further ado, I'm going to pass it off to Gregg. And Gregg, thank you again for doing this tonight.
You bet. Thanks for the invitation, Corey, and the introduction. I'm going to go ahead and queue up some slides and we'll get going. And while I'm pulling that up, thanks to everybody for taking some time on a Thursday evening, and hopefully I can provide you with an interesting approach that we are pursuing at CSU to consider how we can develop a pan-coronavirus vaccine to address the next coronavirus pandemic. So is that presenting correctly, Corey?
Yes, you're good.
And can you see my cursor?
Yep.
Perfect. All right, so here we are in the middle of the pandemic, just hoping to get to the end of it as soon as possible. And amazing that here we are with 7 presentations on this topic. Who thought we would be here? Actually, a lot of people thought we would be here, and that's the point. It's not a big surprise that coronaviruses have emerged again in humans, and that they can cause this big of a disruption in our lives and this much morbidity, mortality, and just chaos with the economy. So, how do we look to the future? And how do we avoid being put through this again? And I'll be focusing on a pan-coronavirus vaccine primarily. And here is just set up for what you can expect as an overview. First, really just want to point out that coronaviruses are present in many different species, and then really address 4 key questions. So first of all, why do we need a pan-coronavirus vaccine? Why not use the current COVID-19 vaccine strategies that we are learning a lot about and are all hoping to get immunized with? And what are the challenges? So when we think about generating a vaccine that can protect us against any coronavirus that may emerge, what do those challenges look like? And then what are we doing here at CSU? So just a reminder, a lot of people know this, but it can be forgotten that coronaviruses have been around a long time and that they infect a lot of different species. Certainly we're aware of the viruses in humans that include common cold, and then the 3 big ones that have emerged over the last 20 years and really cause concern, and then obviously a pandemic now. So SARS-1, MERS, and SARS-2. But the coronaviruses that occur in poultry and cattle, pigs, dogs, they've been around quite a while. They cause different clinical manifestations, bronchitis, enteritis. And then we hear a lot about bats because we know they can be carriers of coronaviruses. It's interesting that often they're asymptomatic and that maybe they are carrying the common ancestor of coronaviruses that have spread through other species. And then cats as well are part of the party here where there's 2 interesting manifestations of a common coronavirus that cause enteritis or can mutate and cause a deadly disease called infectious peritonitis. So there are some common features of coronaviruses regardless of species, and these are generalizations, but they hold true typically that these are viruses that circulate in populations, they enter a population and continue to circulate. They then persist, right? So they're not eliminated from the populations. They can survive in the environment, and so they continue to churn. They're highly contagious. And then there's this interesting association that they're typically mildly or symptomatic or asymptomatic in young. Our treatment across species has typically been symptomatic. So antivirals against coronaviruses have not been widely used or widely developed. Obviously, we're scrambling to try to address that right now. Clearly there's zoonotic potential, and that's how we get here. So not only zoonotic potential, so the ability for the coronavirus to move from animals into humans, but the ability for the virus to move from one species of wildlife to domestic species of animals, for example. And then unfortunately, across the board, there's just very few vaccines that exist. So, I mean, this is really a remarkable time now as we're seeing the emergence of an application, quick application of these vaccines. Into the first question, why do we need a pan-coronavirus vaccine? Well, we have to step back for a minute. We're immersed here in our current pandemic, but let's remember that coronaviruses are not new to humans, And this graphic really shows how long this has been going on. So, and this is just what we can estimate given the technology and samples that we have to look at right now. But if we consider one of the coronaviruses that causes the common cold, we know it's likely it came from bats. But this emerged into the human population maybe as long as 900 years ago. This is just a guess, but it's been a long time. And then similarly, another virus associated with common cold emerged around 300 years ago or so. And so it goes. You can look down the line and see the species that the virus probably originated in, and bats and rodents are most likely. The species that it passed through, so llamas, alpacas, other ruminants here, or maybe civets in the case of SARS-1, camels in the case of MERS. So these intermediate hosts then that pass the virus on to humans, and you can see the timeline. Just want to point out this one that you may or may not have heard about. It emerged about 3 or 4 years ago out of bats into pigs, causes a significant clinical syndrome that's characterized by acute diarrhea. And you know, this is worrisome. This was just published last year. It's known that this virus can infect and replicate in human cells. So, you know, here's a virus right now that we know about that could potentially jump into humans. And I just want to take a second to point out that this is a large area of interest in research at CSU. A number of key faculty that are involved at looking at what species can be infected with coronaviruses. Can they pass those viruses on to humans? Can those species act as reservoirs potentially? So this is an active area of research here at CSU. So getting back to this typical, you know, strategy of coronavirus to use perhaps bats most commonly, and then pass through an intermediate species here, and then jump into humans, and then potentially cause very serious disease and now pandemics, we have to ask these questions. Everybody's asking these questions. How do we prevent this whole cycle of transmission of the virus? How do we detect viruses that could make this jump from one species to the next? And then how do we control that whole situation? Again, these are topics that have been of the interest to researchers for quite a while. I'll say that they've been obviously not funded as well as we would like, or we would be better prepared now for what we're currently experiencing. But even though we're going to focus the rest of this talk on how do we develop a pan-coronavirus vaccine, obviously there's a lot, many more different aspects of how this has to be tackled in a comprehensive way. We have to understand this human-animal interface. We have to identify transmission hotspots and increase our surveillance to understand what viruses are in the wildlife, what are we seeing passed into these intermediate species, so that we have a heads up of what might be coming. How can we increase diagnostic capacity so if something does jump into humans or does jump into a domestic animal, that we can spot that early on before it spreads beyond just an initial outbreak into an epidemic and then a pandemic. And how do we build public health systems that are going to be able to respond to this and control it locally, again, before it spreads more broadly? Certainly having approved antiviral drugs would really help. So if we had drugs that we knew had broad activity against coronaviruses, then we could also apply those to minimize the mortality that could be associated early on in these outbreaks. And then lastly, the pan-coronavirus vaccine. I wanted to take just a minute because there was a couple of questions that came before the seminar tonight, some questions about what are we doing? How can we achieve some of the things that I had on that list of the previous slide? And there are things that are going on and pretty big projects, as a matter of fact. One of them was funded through USAID. It was called PREDICT. It existed for a number of years, and their mission is right here. They sought to reduce the pandemic risk by promoting global health. There was a number of elements here that were also on that list on the previous slide. This was the funding for this program was terminated in 2019 by the previous US administration. There's another big project that's going on, the Global Virome Project. It's really interesting. Here's the website if you want to take a closer look. Again, looking at a lot of these elements of detecting, containing, preventing. This has a much broader support to it and is ongoing. So second question, why not use current COVID-19 vaccine strategies? So we have to take a step back and think about generally what are the obstacles to global vaccination? And there are a number of them, but I list 4 here. First of all, can we manufacture it? Can we manufacture it in sufficient quantity that we can actually immunize every human on the planet? And number 2, can we get that vaccine to those people, wherever they live, whatever conditions they live in, whatever the socioeconomic status, the healthcare infrastructure, et cetera. And related to that, what's the cost? And then finally, does it work, right? So these are all important obstacles. And so we can assess the vaccines that we currently have and apply these questions, consider these obstacles. And so here are 4 vaccines that we're hearing a lot about each day. We know about the Moderna vaccine, the Pfizer BioNTech vaccine. These are both RNA vaccines. What's the problem here? So one, again, that we have heard a lot about is that they have to be stored at very cold temperatures. And this is problematic. So we see the challenges of distributing a vaccine like this, even in a highly developed, wealthy country like the United States. We can look at other examples of vaccines that have been used globally. For instance, the human rotavirus vaccine, which it has been applied broadly, but does require just regular refrigeration, just a regular refrigerator. And Doctors Without Borders point to that simple fact as being their greatest obstacle to fully deploying that vaccine. So any sort of cold chain really is a challenge on a global delivery strategy. Cost, again, so this is a very expensive vaccine. Low-income countries are not going to be able to afford this themselves. They would have to be supported, supplemented to actually deploy a vaccine of this cost. And if you look at the vaccines that we have been successful in global distribution, they're often less than $1 per dose. The closest we get here is the Oxford AstraZeneca vaccine at $4. So there are some issues here. A global vaccine needs to be inexpensive. And then what I have circled here is a viral vector, and I'll explain more about that here in this slide. And this comes right from the Oxford website, and this is how they made this vaccine. So it started with a chimpanzee adenovirus. Why choose a chimpanzee adenovirus? And the reason is because most people have not been exposed to that adenovirus and they don't have antibodies against it. They have taken this virus and then engineered it to take the SARS-CoV-2 spike protein, insert it into that adenovirus, and then use that. That's the vaccine spike protein from the virus, and then the body produces antibodies against it. Okay, so that's the goal of the vaccine strategy. The problem is that people also make antibodies against the adenovirus, so against this chimpanzee adenovirus. So the concern is, and the problem ultimately turns out to be, you may not be able to use this more than once effectively in an individual. And if there are populations that have been exposed to this virus, the vaccine may not work in that population. So that's just sort of an overarching challenge with the adenovirus strategy. So just wanted to describe that. All right, so all of these vaccines that we currently are using under experimental use authorization and those that are going through the approval process right now are targeting the immunity in a similar way. So this slide shows, first of all, some real pretty picture of the coronavirus itself. And then here, one virus particle in association with a host cell. And you can see it setting down on that host cell. And if we look at a cartoon of what's happening there, here's the virus, here's that spike protein. And that spike protein reaches out to contact the host cell receptor, which is called ACE2. So this is how the virus attaches to the host cell. And the goal then of these vaccine strategies is to use that spike protein, inject it into people in one form or another so that people make antibodies against it. And that antibody sticks to this spike protein. So this is the spike protein that looks different here. It's associated with this virus. So the antibody sticks down to what's called the receptor binding domain, and it keeps it from binding the host cell. So that's a great strategy. Keep the virus from actually contacting host cells, avoid infection. Perfect, works great. The problem is that there's a lot of variability in spike proteins across all the coronaviruses that we know about, and almost certainly the ones that we don't know about that we're trying to protect against. So, and we're hearing about spike protein variability right now in the news. It's causing a lot of concern. But if we want to make a vaccine that's going to cover all the, at least a broad swath of coronaviruses that could emerge and cause problems, we have to take this into consideration. So, here in this part of this slide, you can see how each of these coronaviruses compare in terms of the percent of similarity to the spike protein of SARS-CoV-2. So the closest relative at 75% is SARS-CoV-1, and then these others, it's dramatically less. So now we have a problem. So what we're currently targeting with our COVID-19 vaccines is quite specific for COVID-19, and envisioning a spike protein that could be incorporated into a vaccine that would protect across the board when there's so much variability becomes a big part of the challenge. And looking at this idea of mutants, so that's variation from one virus to the next. What we're also seeing, and we're thinking about how we're going to deal with, is the emergence of mutants of SARS2. So These are the spike protein. It's just laid out in a linear way here with different domains. In orange here is this receptor binding domain. And then the different variants that are mutants that have popped up. And these arrows show where these mutations have occurred, just to emphasize that, again, this is a part of the virus that mutates quite a lot. And how does this affect the immune response? Over here in this graph, we can look at how antibodies against the wild type virus may or may not work against these mutants as they emerge. And you can see in some cases it works better, in some cases it doesn't work as well. So the concern is we could have an emergence of mutants that are no longer protected. We're no longer protected against them by the existing vaccines. So there is a limitation if we just focus on the spike protein and the receptor binding domain, that any virus that crosses that species boundary, infects humans, can ultimately mutate and get around that immunity. So to summarize, why not use the existing COVID-19 vaccines? There are some reasons. They are somewhat challenging to manufacture. They are high cost. They require this cold chain. It's sometimes very cold. And then this focus on spike protein may not work necessarily. You know, some of these things we may be able to overcome, especially the cost and the manufacturing part. But whether that solves all our problems, we need to consider other options. So if we look at this with fresh eyes and say, all right, what are the challenges to this pan-coronavirus if we're going to approach it in a different way? Well, you know, the challenge here is the challenge that we see no matter what vaccine we're trying to develop. It's always the same. We have to choose an antigen. So what are we going to put in the vaccine that we're going to induce an immune response that's going to provide protection? What platform? So we've talked about RNA, we've talked about adenoviruses, there's others. Ultimately, we've got to provide efficacy. So we need to demonstrate that our vaccine is able to produce the desired effect. We want to protect people against infection or disease. And it's got to be safe. So safety is critical. So jumping into those challenges, what are the challenges? First of all, how do we identify common antigens across all of these coronaviruses? There's three ways we can look at this, and maybe there's more, but three that I'm going to point to today. We can look at the genetic sequence of the viruses. We can look at commonalities in the structure. And we have to consider viral evolution here. So this cartoon lays out the whole genome of these three coronaviruses that have emerged over the last 20 years in the human population. Their overall genome is laid out fairly similarly, as you can see. So if we take that and we go to the next level and look at the genetic sequence, we can go bit by bit and really line up the genome sequences from viruses from bats and civets and pangolins and camels, and really say, all right, what are conserved areas within this sequence? And we can identify those across the entire genome, and those may be targets for us to explore.
Greg, before you move on, we have a question. It says, how different is the 95% effectiveness of vaccines compared to the infection rate without vaccines?
How different is the 95% effectiveness? Can you repeat the rest of that, please?
It says, how different is the 95% effectiveness of vaccines compared to the infection rate without vaccines?
I'm not sure I understand that completely, but a 95% protection, and those numbers were based on the circulating virus at the time. We have to think about that. Meant that 95% of people who had the vaccine and then were exposed to the virus were presumably protected. As the virus mutates, how does that percentage of protection decline? Well, we don't know. That's something we're actually studying in real time now. If we didn't have the vaccine at all, well, then we're back to square one. Everybody who is exposed has a much higher likelihood of being infected. I don't know if I answered that well. I'd be happy to elaborate on that if I didn't address the question well. Thinking about structure of the different proteins that comprise the virus. So structure and function are related. The virus looks like it does for a reason. And part of that is laid out here again, looking at the spike protein. And this is just a different way to look at it. I've showed you a number of pictures of the spike protein. What I want to point out here is that there are a couple of areas, and they're here, S2 prime and S1, S2. These are regions that are cleaved by enzymes. Cleavage at these sites is very important to change the structure of the virus, and it's related to its function. And I'll explain more about that. This is actually the spike protein from the feline coronavirus. It looks very similar to the human coronaviruses. And the spike protein here is laid out, and you can see all the different domains, the receptor binding domain, and then these 2 points where enzymes can actually cut this. So what does that mean for the virus? Here again, here's a virus. This is the ACE2 receptor. The virus engages that receptor. This is the host cell membrane. So again, this is how the virus engages the host cell. And then what happens next is really complicated, actually. Those two sites that I mentioned, S2 prime, S1, S2, those are cleaved by enzymes. And what that does is change the structure of that spike protein. So it draws the virus close to the host cell membrane and allows the virus to fuse with the host cell. And that's essential. That's essential for infection. So what structural components that are critical to the success of the virus can we target with a vaccine strategy? And we can do the studies, and this is just one that I pulled up that showed that if you eliminate those cleavage sites, those enzyme sites, that this virus is no longer able to infect human lung cells. So that presents an opportunity for us. The other thing that we have to think about, again, is virus mutation. And I just wanted to point out sort of relative mutation rates so we can put it in perspective. And so here we can look at the whole size of the genome and the different categories of organisms, and then the actual mutation rate here. And if we look at us, humans, other mammals, eukaryotes, we see that the mutation rate is actually quite low. About 1 in 50 million bases might mutate during the replication of eukaryotic cells, or I'm sorry, 50 billion bases. So very, very rare event, relatively speaking. On the other hand, if you look at RNA viruses, so coronaviruses are RNA viruses, about 1 out of 50,000 bases will mutate during the replication of an RNA virus. So much higher mutation rate. That's why this becomes a challenge. This virus can adapt to us much quicker than we can adapt to it. And we see that. So we're hearing about it, and this slide really shows that. So if we're looking here now at the time of a virus outbreak and going through these waves of virus spreading through the population, initially we have infection of very few people, and this is what that founding virus looks like as it begins to move from person to person, and we go through these waves of infection. We see accumulation of mutations, which could be adaptive, meaning that it helps the virus to maybe transmit more easily, or it could be neutral or deleterious. So, nothing happens, or it's a dead end for the virus. And then over time, and we're seeing this now as we sequence more virus in people, the accumulation of these viruses, and then the predominance of different viruses that are circulating in the population. And you can recapitulate this in the laboratory using cells in Petri dishes, putting in one kind of virus, and then that will infect these cells. You take the virus that replicates, move it from one Petri dish to the next, and you can see the accumulation again of these mutations and the emergence of a very different virus as compared to what we started with. So we have to anticipate that. That's going to happen, continue to happen with SARS2. It's going to happen with the next virus. So, a question here is, can we guide the immune response in a vaccinated population so that it can drive the evolution of the virus towards a less virulent form? And this ultimately happens naturally. So if we, excuse me, if we didn't intervene, this virus would go through humans around the globe. It would kill a lot of people. The people who survive would establish some immunity and then begin a coevolution with the virus. So that the virus would still avoid the immune response, but we would reach an equilibrium where we don't see people dying anymore, and the virus just churns in the population, like I mentioned before, just like our coronaviruses associated with common colds do. So, what we're talking about is using the immune response to basically herd cats. Ordinarily, the virus is just running all over the place, mutating, and it's out of control. Can we intentionally use the immune response through vaccination to herd the cats? So that's an interesting idea. Okay, so choosing a vaccine platform, and I'm going to focus here on what we're doing at CSU to address these questions. And here's the virus, here's what we want to protect against. These are the vaccine platform strategies that we have. There are others. We've talked about RNA or DNA vaccines a bit, and we talked about viral vectors, the adenovirus. What we are focused on in our current strategies, and I'm going to elaborate on this, is two approaches. One is recombinant bacterial vectors, and then the other is a whole inactivated virus strategy. So starting first of all with one of the bacterial vectors, and this utilizes recombinant BCG. And BCG, that's what this stands for here. These are the two discoverers of BCG, and this is actually a tried and true vaccine strategy that's been used for quite a while against tuberculosis. So it's been put into more than 5 billion people, 5 billion doses given to date. And so we know what the safety record is, and it's very good. We can make these bacteria that express the coronavirus proteins in it, so it's recombinant. And we can potentially use this then as a vaccine against pan-coronaviruses. So, these are more details about why we might want to use this. It's very effective at a single dose at inducing quite a broad immune response. It's very inexpensive. It's easy to produce. It doesn't have to be refrigerated. So these are real advantages thinking about delivering this globally. So where are we at with this? So Mary Jackson has been driving this work and has made recombinants and is in the midst of preclinical testing with these recombinant BCG bacteria currently. This is really interesting, though, and I wanted to elaborate just a bit because there's been observations made as we're studying the current pandemic, because there's some inherent ability of BCG to help us, help people who've been vaccinated for TB with BCG to have some resistance to infection with SARS2. And this map shows here where BCG is used to vaccinate against TB across the world. So in orange, these are parts of the world where it's routinely used. Purple, it's used less broadly, and then red here in North America and Italy and in the Netherlands, it's not used at all. And then this graph here shows a BCG index, so countries that you do more BCG immunization than those that don't, and then the number of deaths due to COVID-19. And you can see that there is this correlation that those countries with an extensive vaccination with BCG are experiencing less infection and death. The second strategy here is employing recombinant lactobacillus acidophilus. You may know this is a probiotic, so it's something that's found in food and is used for a variety of general health purposes. And as such, it's generally regarded as safe by the Food and Drug Administration. So the safety profile, again, is very good. It's recognized that it has a vast potential as an orally delivered vaccine. So that's one of the reasons we want to explore this. It's also easy and inexpensive to manufacture. Doesn't require the cold chain. Since it's delivered orally, you don't need a needle, so you don't need a trained medical infrastructure necessarily to deliver this vaccine. This is a project that's ongoing in my laboratory, and we've developed a number of recombinant lactobacillus acidophilus vaccine prototypes, and are also in the midst of preclinical studies and other work not in our laboratory on the manufacturing of this is also underway and is really well understood. Okay, lastly is something that if you've attended other talks in this series, you may have heard about whole inactivated virus, and specifically the Solovax vaccine that Gregg Goodrich and many others are working on at CSU. So, this uses a whole inactivated virus. What that means is you grow up the virus, you inactivate it so it can't infect. And that's done with this very interesting photochemical approach that Gregg Goodrich was instrumental in developing. And then you purify that inactivated virus, and then that's injected as a vaccine strategy. So, there's a lot of whole inactivated virus vaccines that have been used for a long time. So it's tried and true. if we think about using both SARS-1 and SARS-2 in MERS, combining those together, that gives us this multivalent approach. And so how would that work? Could that induce a broad enough immune response that it could protect not only against those 3 viruses, but maybe also against other coronaviruses? So again, this is relatively easy and inexpensive to manufacture. And the results from this approach could really help inform the other 2 strategies that I just mentioned. So where is this at? So there were some questions ahead of this talk wanting to know where are things at? And I talked to Ray just before, and this is really moving along. So it's pretty exciting. They're into a second phase of the preclinical studies and really looking at the scale up of manufacturing and how to stabilize and validate the vaccine for larger scale manufacturing. So it's moving along. So that's exciting. So what we're talking about here is this sort of approach, really using what we're learning right now, what's in the literature, and what we'll continue to learn from ongoing work while we're in the midst of this pandemic, to use informatics and studies that are happening both in natural infections and experimental infections, and then work that's going on in the laboratory to inform our choice of antigens to include in these vaccines, that we can test in animals, make these constructs, look to see whether it works, and then iterate this process. So see what works, what doesn't work, what's happening with the immune response, what are we learning around viral evolution, and use that to help us as rapidly as possible advance some vaccine candidates. There was another question that came before hand that asked about antibody enhancement, because that's a very common problem that's been encountered in developing a vaccine against the feline coronavirus. So veterinarians and people who've had cats that have suffered this disease may be aware of this, but the problem here, and this really gets to the safety component, the problem is that there is a potential that if you immunize an animal, an individual, that that immune response could actually enhance infection. Obviously, that's exactly what we don't want to do. So, this is something, as a veterinarian, as someone who's worked on the feline coronavirus, and we're also working on a feline coronavirus vaccine, this is top of mind for us. Clearly something that we have to attend to. Ray has not seen this with his Solovax vaccine. So that's excellent. It's something we have to address. And these are just a couple of publications, one from Cats here that sort of points out this potential problem. And also, to point out that this is not being not considered by everybody who's looking at vaccines against COVID-19 right now. So, this is a huge project. It takes a lot of people to put their heads together, have that expertise. It's really interdisciplinary. And here are a number of people here at CSU that are involved in this. We're excited about what could be accomplished, because over this last year, we've worked together really focusing more on SARS-CoV-2, but really anxious to turn towards addressing what we know is going to be the next coronavirus that will emerge, will jump into humans. Can we be part of a solution with a pan-coronavirus vaccine strategy? And I just want to acknowledge all of them. They're fantastic people. And like I said, there's others that are involved. But sort of going back to the very beginning, we've focused now on coronavirus vaccines, pan-coronavirus vaccine, but importantly, we've got to attend to environmental health, human health, animal health, and this whole concept of One Health, which is really our philosophy. We can't expect to solve all of our problems with a vaccine, with an antiviral. We've got to take care of everything. And finally, it's our great privilege to be here at CSU and land grant university, where we can do this work that we of course hope benefits Colorado and the nation and the whole world. So that's it. That's all I have. I thank you again for your attention and for attending, and I am more than happy to take any questions.
Cool. Greg, there is a question that happened earlier on. I just wasn't able to stop you. And feel free, anybody else who has questions, to write them as we're answering these. This question says, the UK virus strain has now been found in Weld County. Weld County and the JBS slaughterhouse was a prime source for infection for quite a few months. Slaughterhouses and meatpacking plants have been found to be places of high infection. Not only the animals in these places under tremendous stress, a prime ingredient for the jumping of virus from species to the next. But the temperatures at the meat packing plants are prime for the virus growth. Would it be better to start focusing our attention to the treatment of animals in high density locations? That was long, I'm sorry.
There's a lot to unpack there. No pun intended. But we know that there's high suspicion that in the so-called wet markets, where we're bringing animals and people together in that slaughtering environment, there's a chance for transmission of coronaviruses. So, they're not really necessarily looking to see whether the animals that are brought in have coronaviruses. We can test animals. So the animals that are coming into slaughterhouses, we can certainly look at them and we can apply controls there. We have to look at the conditions of people that are working in those environments. Are they adequately protected? Are we doing everything in those environments to prevent the transmission among the workers? For sure. It's interesting that that the UK virus is popping up there. I always ask the question, where did it really come from? Did it come from the UK and spread in the population here? Or did the virus just evolve in a similar way in an environment where there's been a fair amount of transmission and replication, and come to the same conclusion that this is a mutation that works for it? So there are a lot of issues to address, both at the operations, at high density animal agriculture, and how we deal with that going forward. Surveillance, again, complicated and super important issue.
Cool. Okay, next question is, which animals are best for the viral studies?
Yeah, so there's maybe not an agreement, and I think, as always, the question, the answer is, what's the question? So hamsters are used for vaccine studies. There are also mice that have been generated. They're transgenic animals that now carry the human receptor for the virus. So that's one strategy. There's also a virus that's been modified so that it infects mice more easily. It depends on what the question is. I don't feel that there is a consensus in the scientific community on that right now. There are always caveats about animal models, and you just have to keep those in mind as you're doing the studies.
Next question is, do you know what information the World Health Organization is lacking from their recent visit to China to investigate the source origination of the virus?
Yeah, so I'm not sure that that detailed report is out yet. And whether it's come out in the last couple of days, I don't know. But um, just hearing what they were able to get their hands on there, they were able to get to quite a bit, but they didn't see some of the primary data that was generated. Um, it's a politically sensitive issue, and I don't think they're talking too much about the details of this. They're hoping to get everything that they asked for. I don't think they necessarily did. I think they're happy with the progress they made, but certainly they're hopeful to see more.
Awesome. And that are all the questions that I have. So if there are no more questions that come in, feel free. You guys can go ahead and log off early. You get 4 minutes left of your day. Oh, hold on. I just got another question, I think. The zinc seems to be a common denominator in several of the supposed treatments. Zythromax, I don't know these names, ivermectin, and by itself. Can treatment be as effective as vaccinations?
So I I always say that to really deal with an infectious disease, you've got to have three things. You've got to have a diagnostic test. You'd really like to have some sort of therapeutic modality, and you need a vaccine. That's going to be what we rely on from a medical point of view. Um, we really need antivirals that work to complement vaccine failures, because there's going to be vaccine failures. So yeah, I think we have to vigorously pursue antivirals.
Cool. And then another question just came into, do you know how remdesivir works against the virus?
Yeah, I had a lot of high hopes for remdesivir because it showed some pretty good promise in cats infected with coronavirus. It seems like it hasn't been as dramatic in humans. It does target specific required processes that the virus needs for replication. So it's quite virus targeted. So rather than a general process that would also affect the host. So that's a very nice part of that. It is virus targeted, and there's other antivirals out there, and just a ton of work that's going on to look at individual antivirals and then combination antivirals to see if we can hit on a strategy that would really provide that option for doctors to go to when they need it.
Cool. And then another question, do you have a favorite antigenic target, something highly conserved?
Yeah, all of them. It's going to take a lot. So because we're targeting such a wide range of coronaviruses, we have to go with a kitchen sink approach. Everything and the kitchen sink. I don't think we're going to hit on just a couple of weak points in the virus that's going to provide us the outcomes we're looking for.
Gotcha. Okay. And with that, that was the last question. So thank you again, everybody, for showing up tonight. We hope that this was a fun and educational opportunity for you. Gregg, thank you so much for doing this event. And we will be sending out a link after this with a link that goes to our YouTube channel with the recording of this. So if you ever need to look back on it or rewatch the video, you can check it out there. And with that, thank you again for everybody being here tonight, and go Rams.
Thank you all. Have a good evening.